Genetic engineering is the modification of an organism by artificial means. This often involves the transfer of a specific gene from one organism into another. When the transfer occurs, the resulting organism is called transgenic, or GMO. GE offers unlimited opportunities for creating a new combination of genes which at the moment do not exist under natural conditions. Selective breeding of plants and animals through natural variations have been done for centuries, but GE is different from selective breeding. This technology uses recombinant DNA to help modify the genetic properties of an organism. If genes are viewed as software and they program the growth, drive, development and function of an organism, but when the software is changed in a controlled, precise manner, it is possible to change the characteristics of the organism. The foreign DNA or gene is introduced into the host organism in such a way that the total genome of the host cell is unchanged, all except the manipulated genes. DNA can be isolated from cells from different organisms and they can be fragmented into groups of one or more genes. Fragmented DNA can be coupled to another piece of DNA and then passed into the host cell. The host cell then formed genetic properties and chemical abilities which were unattainable by conventional ways of selective breeding. This technology lets the breeder to select a particular gene required for desired characteristics and modify only that gene. The majority of GE crops that are grown today are engineered to be resistant to pesticides and herbicides, so the crops can withstand being sprayed with weed killer. There is a claim that GE crops use fewer pesticides than non-GE crops, but in reality, GE plants require more chemicals because weeds can become resistant to pesticides, which leads the farmers to spray even more onto their crops. This leads to polluting the environment, meaning a higher exposure level to toxins, and this creates a safety concern for farmers and farm workers. There is research on ways to genetic engineer animals. For example, Atlantic salmon have been genetic engineered to be twice the size as wild salmon. Chicken have been genetic engineered so they cannot spread bird flu to other birds. There are no genetic engineered animals that have been approved by the FDA to enter the food supply. To make a genetic engineered animal is to get healthy egg cells removed from a female animal and they are fertilized in the lab. A gene from another species is identified, isolated and lastly cloned. The cloned gene is injected into the eggs, then they are surgically implanted into the female and the embryo undergoes a normal development. It's hoped that this process will provide a rapid but cheap means of genetic enzymes, proteins and increased production of meat, wool and other animal products. rDNA allows scientists to produce products that were previously unavailable, e.g. insulin. Until 1980s, the only source of insulin came from animals and the supply was never high enough to meet demands, so this drove up prices. The reason for this is the animals were slaughtered for meat and other purposes. In 1982, the FDA improved the sale of insulin, which was produced by genetic organisms. Since then, there have been several additional products that have been made with rDNA, e.g. human growth hormones. Two products that were first to be prepared from rDNA is insulin and human growth hormones, which were cultured in E. coli bacteria. A number of vaccines are now commercially prepared from host cells, but there was a time when the natural but small amount qualities of the disease was injected into someone just so the immune system was activated to make vaccines in the future. The downside was some of the patients ended up with the disease. Industrial microbiology. The production into microorganisms has a wide range of products, e.g. human and animal proteins. In medicine, there are better vaccines and hormones. In agriculture, there is improved plants and animals for production, quality, taste and safety. In the environment, a range of benefits such as pollution control can be expected. food industry has improved strains of bacteria and fungi which are influencing traditional processes, e.g. baking and cheese making. Understanding the working concept of our DNA technology requires a good knowledge of molecular biology. There are basic techniques of expressing the foreign DNA in a host cell. This is isolating, cutting and joining molecular and inserting into a vector. DNA can be cut using enzymatic or mechanical methods. Specific restriction enzymes are used. It is possible to recognize specific target-based sequences in double-strand DNA. Large number of different endonucleotides have been extracted and classified from a wide range of microbial species. They are named according to the species from where they were first isolated. They can distinguish between DNA from their own cell and foreign DNA. This allows a break, opening the DNA into shorter fragments that contain a number of genes which determine the enzyme used. The fragments can be separated from each other on the basis of molecular weight. DNA fragments that have blunt ends or corrosive overlapping ends can be joined together. Molecules that have DNA being inserted into them have been termed DNA chimeras because of the analogy with the chimera mythology. Vector molecules have been developed as the vehicles of gene transfer, e.g. plasmids. Vectors exist within a cell in an independent or extra chromosome. They are capable of entering the host cell and replicating with it. Vectors should be small, easily prepared, and must contain at least one site where the foreign DNA does not destroy essential functions. Plasmids offer the greatest potential in biotechnology, and they have been found in a range of organisms, e.g. bacteria and yeast. In the 1970s, a small group of recognized molecular biologists met to express the concerns of the safety of the technology. In a published method, it was focused on the potential but high risks that are associated with rDNA and how it should be managed. The NIH published the Guidelines of Research Involving Recombinant DNA Molecules after the conference. This sent a message to the scientific community and federal government departments. The NIH was concerned about the risk scenarios. The guidelines became the standard regulations for most of the world. 30 years later, a wealth of scientific peer reviews had looked at the risks concerned and stated that the fears suggested are largely groundless. GE was first experimented with microorganisms in the 1970s. Many molecular biologists believed that the process was unsafe and that the manipulated microorganisms should be contained and prevented from being released into the environment. The fear is GE microorganisms could escape from the labs into the environment with unpredictable consequences. Guidelines were established to ensure safe working practices and levels of containment based on potential hazards. Many of the regulations have been relaxed over time to a low-risk system. Medical products is manufactured in a large-scale containment fermentation process which involves specific genetical manipulated microorganisms. The final products are free from genetically manipulated host organisms. The system works well today. There has been no health or environmental problems. Conventional genetic techniques have been accepted worldwide and haven't caused any problems. Their two main advantages are the introduction of genes can be controlled with greater precision and prediction than previous methods. The introduction of genes into unrelated species, which is not possible to achieve by traditional methods. The safety of human food supply is based on the concept that there should be no harm that will result from its consumption. GMO foods/ingredients must be considered to be safer than their traditional counterparts before they can be recommended as safe. The most practical approach to determine the safety of food that's considered new food as substantial equivalent to conventional food products where they are and intended use/exposure. There is a social price to pay with biotechnology, particularly in developing countries. For example, the development of sweeteners is already reducing the traditional sugar market for sugar, disrupting these economics. Developing countries will suffer more than industrial nations. Many developing countries are making investments and progress in relevant biotechnology. A UK report identifies some of the main ethical concerns relating to transgenic organisms. Human genes to food and animals, genes from animals whose flesh is forbidden by certain religious groups for use in food, animal genes in food plants which could concern vegetarians, use of organisms containing human genes in animal feed.